Hi, I am MD Navid Akbar, and I welcome you all to the presentation of our work titled Mapping Motor Cortex Stimulation to Muscle Responses, a Deep Neural Network Modeling Approach. I am from Northeastern University, Boston, Massachusetts. Let us now dive into the paper itself. Introduction. So, stroke and other neurodegenerative diseases have been a major cause of loss of motor or limb control in human beings. So, as a remedy, a recent study, what we have been doing is investigating the use of a non-invasive intervention called transcranial magnetic stimulation, which actually induces electric fields into the human brain and helps to restore the motor control. To reliably apply this therapy, we need to first understand the healthy cortical control of multi-muscle coordination and develop a corresponding model between the brain and the muscles. So this is what a TMS coil and the effects it has on the brain looks like. These are the E fields generated by the TMS stimulation. This is what we are calling a neural firing input or a neural thresholding, which we are going to talk about soon. And this is the motor cortex responsible for motor movements or muscle movements. Methods. First, we want to build such a model by applying TMS at 4 different intensities or resting motor thresholds to excite 15 muscles in the hand. A finite element simulation to reconstruct those electric fields, an empirical neural response profile, yes or no, we might or might not use it, electromyograph records of motor evoked potentials indicating the muscle activations, and finally a machine learning technique, which is a deep convolutional neural network containing an autoencoder for obtaining low dimensional representations and a corresponding deep network mapper. So we have explored different CNN models based on neuromuscular control, as we find in neuroscience, different types of input, meaning direct E fields or this use of neural response, and the complexity of the network. Then the best CNN model of the ones we have tested, we recommend it for M2M, what we call M2M net, the motor cortex to muscle network. So this is what the entire problem statement from a very bird's eye view look like. What we are trying to solve basically is this mapping between the brain and the muscles. Now this is how our system block diagram looks like. So first we have the round bit, which is a 64 cube representation of the motor cortex or the electric field distribution in the motor cortex. So the modeling choices are marked by these green squares. So first, whether or not we want this high pass filtering or this neural firing conversion. So this has been empirically derived by studies in macaques, but we want to see if this works as well in human brains as well. This is the input to the network itself. We have some convolution layers, and we go to a lower dimensional representation to a 16 cube, which is the latent space, and then we again go up back to the 64 cube representation, and by optimization of this error function in the autoencoder, what we are trying to do is get a reconstructed input that looks as close to the input as possible, which means the latent space that we have for the 16 cube is a unique identifier of that input. So one question is, why are we going to this low dimensional representation? Yes, so the major reason is we want to decrease the number of input parameters, because in as you might understand in medical data, the data is actually scarce, so we have to make meaningful use of the less number of training data we have. So going to a low dimensional representation is quite paramount. So these low latent space representations go to a second CNN network, what we're calling the mapper. It has further convolutional layers, 2 fully connected layers, and finally another fully connected layer, but this is This again has choices, just like here this previous layer, which has 3 or 6 layers. So in the final, we only have one fully connected layer, but it could be direct mapping to those 15 muscles in the hand, as we mentioned, or a non-negative matrix factorization. We have found 9 to account for the lowest variance. Maximum variance accounted for, and both representing the network first trained to the synergies and the differences trained to the net direct. That way we have both. Now the mathematical model of the system looks like this. It might look a little intimidating at first, but these MIs are our output resultant muscle activation vector corresponding to a single trial, meaning for every stimulation. The F here represents the neural network, the CNN, the M2M net, D for the direct connection, S for the synergy, and for the both network, as I have mentioned before, we're first training them to the synergies, and then the difference or this error epsilon, we are using this and we are training that with the direct connection. So there is a D star. This is another direct connection, but the parameters might be slightly different, and we expect to get a lower error epsilon b. So this is the non-negative matrix factorization we were talking about. So B is our basis vector, basis matrix, which governs this transformation from this muscle activations to the synergy activations. And this is our error metric, which is the normalized root mean squared error. As you can see, it's just basically the error squared given by the energy of the trial or the signal itself, and the sum of all taken by root. Finally, here we also take a look into how we have extended the derivations for our network to two popular statistical information criteria, the Bayesian information criteria and the Akaike information criteria corrected. And this is basically like a very top view summary would be, this is based on the fact, the assumption that the errors are Gaussian distributed, and this is the squared error, which is the average squared error given by this expression. Moving on to the results. So we have data from a single healthy subject, as I mentioned, 4 different intensities, 699 stimulations, 3 repetitions of all the sets with a trained test ratio of 9:1. This is how our results look like, and as it might be evident that the CNN models that have the neural firing input actually perform better, evident by their lower errors. This could be due to the fact that the neural firing input causes increased image contrast and possibly aids in better easier feature extraction. Then the 3-layer mappers correspondingly perform slightly better than their 6-layer mapper counterparts in the neural firing input, but with the raw E-field input, it's actually the opposite, which could be due to the fact that the raw E-field input has more, the data has more parameters, less zeros, it's less sparse, so we need more parameters to estimate. So these higher or more complex models are doing better. And again, we will go look into this later again, but these apparently high values of the NRMSE mean and the standard deviations could be due to the fact null stimulations, which we're going to again look in 2 slides. This is the performance versus complexity. As we can see here that the 3-layer both performs the best, as we saw in the previous slide too. But here we can see that even though those errors were comparable, 3-layer both has a good match of errors and complexity trade-off. It's not too complex, it has the lowest error and less number of parameters than this. Probably are insufficient, more could overfit likely. And if we look into the BIC, we see that the BIC tends to favor the models with the lowest complexity, whereas AIC tends to favor the models with the lowest error performance. But again, we must be wary that both of these criteria actually look into the training performance, which is not necessarily an indicator of the test performance of the CNN, and could actually be an indication of overfitting. But still, regardless, it gives an overview of how look into how the performance versus complexity fares. Finally, this is a sample output for the three-layer mappers with the neuron finding as input for 70 test trials. Again, we see that the direct synergy, the error goes down, but the both connection, which takes into account both synergistic and direct connections, actually performs better, and you can look into the subtle nuances that it actually represents it better. And some of these, there are those zero or low activations which actually do drive up or NRMSE, possible explanation of these high values. And at the conclusion, we have again recommended the lowest NRMSE model, which the both model with the three-layer mapper, neural filter input. And again, this result demonstrates that it is very useful to use both empirical or experimental knowledge along with theoretical understanding and underpinning of the corticospinal anatomy, and could help in preclinical mapping. With that said, I would like to thank you all for listening to me, and any questions, please feel free to reach out, and have a good day.